Okay, looks good. So hello, good morning, and welcome everyone to another Rapid Novo webinar. My name is Wei, and I will be moderating today's session. Today's presentation is titled Navigating Intellectual Property Protection for Antibodies, a joint webinar brought to you by Rapid Novo and Venner Shipley. Before I begin, I will go over a few housekeeping tips. First of all, we will be doing a Q&A session towards the end. So if you have any questions, we welcome you to type it into the Q&A chat below. As well, the presentation is being recorded and will be made available to everyone shortly afterwards. Next slide, please. So just a quick introduction of our speakers today. Today, we will have Kirsty Dolphin from Venner Shipley, as well as Monique Seymour from Rapid Novo doing the presentation. Kirsty is a UK and European patent attorney at the Venner Shipley Chemical and Life Sciences team. She has experience drafting patent applications and prosecuting them worldwide and regularly handles oppositions and appeals before the European Patent Office. Kirsty's practice covers inventions across a broad range of biotechnology sectors, such as gene therapy, vaccines, recombinant DNA technology, sequencing and screening methods, diagnostics, and antibodies. Monique is our Senior Scientific Sales Executive for our international markets. She graduated from the University of Glasgow with a Master's in Biotechnology and Management, and she has several years of experience working in the proteomics industry, regularly engaging with scientists in both the diagnostics and therapeutic areas. So thank you for you both to presenting today. Next slide, please. In today's webinar, you will learn why IP is important in the development of antibody-based assets, understand the patent application process and requirements, gain insights on how to strengthen patent applications, and finally, we will be discussing the utility of next-generation protein sequencing, SPR binding kinetics, and HDX-MS epitope mapping technologies in antibody-related patent applications. So I will hand it over to Kirsty to take over the presentation for her section. Thank you very much.
Okay, hopefully you can all see my screen. So just a little bit about Venner Shipley to start with. So we're a specialist intellectual property firm. We cover full range of intellectual property, patents, trademarks, design, copyright, utility models, licensing, SPCs. We have a team of both patent and trademark attorneys, and we also have a legal team who can help with any contracts licensing issues, for example. Wei's already given me an introduction, so I'm not going to go through that again, and I'll jump straight in with what we're talking about today. I'm sure everybody in the audience knows roughly what antibodies are, but just in case not, I'll do a very brief introduction. Antibodies made up 5 out of the top 10 drugs by sales in 2022, and over the last few years have become increasingly important in the biotech and medical space. So antibodies are immunoglobulins comprising a Y-shaped protein with 2 identical light chains and 2 identical heavy chains, which are each folded into constant and variable domains. Each of the heavy and light chain variable domains, VH and VL, have 3 hypervariable regions referred to as complementary determining regions, or CDRs, and it's those regions which provide the antigen-specific binding interface. Switching now to looking at the general requirements for obtaining a patent, and this relates not just to antibodies, but to all inventions. The first requirement is there has to be a technical character and has to be industrially applicable. That's generally a very low hurdle to meet, and antibodies will meet that requirement. Antibody has to be novel, so it can't have been made available to the public at all. It has to have an inventive step. So it has to not be obvious to what we call the skilled person in the art. It has to be sufficiently disclosed. So the invention that you're claiming and the antibody you're claiming has to be reproducibly described in the specification such that someone else can carry out the invention. And it also has to be clear. So the claims have to define the invention or the antibody and leave no doubts regarding the scope of what is actually claimed. In March 2021, the European Patent Office guidelines for examination included a really updated section devoted to the examination of antibody-related inventions. And there's a link to the article that we wrote on our website there. And then minor amendments were made last year. And essentially, they describe how we can claim antibodies, and there are several different ways in which we can do that, and I'm going to go through each of them in turn. So the list is there. So we're going to talk about structure, function, target antigens, target epitopes, hybridomas, and methods of production. And we often use a combination of features. So for example, defining the antibody by both its structure and function was how 55% of patents since 2021 have defined their antibodies. So starting with structural features, and a type of claim you might have is, for example, an antibody comprising the CDRs of seq ID numbers 1 to 6, and you claim the sequences of the 6 CDRs. Or, for example, you could have an antibody comprising a VL domain comprising seq ID number 1 and a VH domain comprising seq ID number 2. So you're describing the entire domain rather than just the CDRs. You can claim variants of the sequences. So, for example, 90% sequence identity to sequence ID numbers 1 to 6, but you will need some data proving that those variants still bind the antigen. We tend to include single CDRs in the claims, although they're not, and they don't tend to be necessarily new on their own, but it can be worth having a claim in there to them, especially for some clarity reasons. You can claim less than one CDR if you have supporting data to show, for example, that these particular CDRs are essential for antigen binding, but others are interchangeable. But typically examiners do want to see all 6 CDRs unless you've got data showing that you can have less than that and the antibody still functions. Heavy or light chain on its own is unlikely to have the binding properties of a whole antibody. So unlikely to get away with claiming that. And sequences are novel provided they're not described in the prior art. But at the European Patent Office, there's no such thing as structural non-obviousness. So just because the sequence is new, that's not going to necessarily give you patentability. The antibody is only going to be considered inventive if an unexpected effect or property is shown. Defining the antibody by the CDRs is definitely one of the most popular ways to define antibodies, and 85% of patents granted in the last couple of years define the antibody at least in part on the basis of the amino acid sequence. So when we talk about unexpected properties that we need the antibody to demonstrate in addition to the sequence being new, and these type of features can help you to demonstrate an unexpected technical effect. There's a whole list of things that could be possibilities. And I listed them here. I'm not going to read them all out in turn, but one that we use a lot is specificity for a new epitope or improved binding affinity. What you will need, however, is experimental evidence to show that you've demonstrated the property that you are relying on, and any associated advantages should be in the application. Otherwise, if you just list, even though it's a new sequence, the antibody will likely just be considered a mere alternative, which isn't going to be inventive. Moving on to functional features, you might have a claim along the lines of an antibody that binds to antigen X with a binding affinity of greater than, or an antibody that binds to antigen X on the surface of the cell and induces apoptosis. And you would include the target and the activity in the claim. if you have any parameters in the claim, they've got to be clearly described and reproducible. And again, you'll need experimental evidence in the specification that is reproducible. It is possible to claim an antibody characterized by the sequences of both variable domains or CDRs with less than 100% identity to those sequences when you combine the sequences with a clear functional feature. So if you want to introduce not just the CDRs, but for example, at least 90% sequence identity, then you are more likely to be able to do that if you include an additional feature such that the antibody is capable of reducing the binding of ligand X to receptor Y, for example, or whatever the functional feature that you're considering might be. We can also look at claiming antibodies by their specificity. So, for example, antibody X that binds specifically to antigen Y. A list here of ranging acceptance by the EPO is listed. So if your antigen is unknown and the antibodies are unknown, then you're likely to be considered novel and inventive. That's quite a rare circumstance these days. Typically, the antigen is known. And if that's the case, but antibodies against it are unknown, it's going to be novel and likely inventive if the antibody is difficult to isolate or exhibits, again, an unexpected property. If the antigen is known and other antibodies against it are known, you'll have to demonstrate that the antibody exhibits an unexpected property over and above those which are already known. If the antigen is known and claimed antibodies are also known, that's not going to be novel. But for example, if there's a new use of the known antibody, that might be novel and inventive if it has a surprising property, for example, if it can be used in a vaccine or another delivery method that's not otherwise known or obvious in the art. So just to summarize, if the antigen is novel, then an antibody against it is usually considered to be novel and inventive. But if the antigen isn't, then the antibody is generally only thought to be inventive if it shows unexpected properties or if it was unexpected that an antibody could be produced at all. And having a generic antibody against a known antigen is not going to overcome those hurdles unless you can demonstrate such unexpected properties. And relatively few patents granted since 2021, only about 5% define antibodies on the basis of the target antigen. Another way we can look at claiming antibodies is the target epitope. So, for example, an antibody that binds specifically to a particular amino acid sequence, if it's linear, or if you have a non-linear epitope, you could define it that it binds to a specific sequence within an antigen defined by a longer sequence listing. Again, if the epitope is unknown and an unexpected property is shown, then the antibody is going to be patentable. And you can define the novel epitope one way or another, and not necessarily by its sequence, but you could define it potentially by its function. And using nonlinear epitopes is okay, provided the target antigen and the disparate regions forming the epitope and their positions are clearly defined in the claim so that somebody else can understand exactly what is meant. And in this situation, it's likely to be necessary to provide data that prior art antibodies binding the antigen don't bind to the particular epitope that you are claiming. Another way to claim antibodies is an antibody produced by a Hybridoma cell line, which has been deposited under a particular accession number. We don't see this very often, but it is possible. And there are a list of, particularly for the EPO, requirements for deposited biological materials that will apply, and particular timings that had to be adhered to, so that at the time you filed your application, your deposit was already made, so that at the time you filed the application, someone else can reproduce the invention. And I think coming to the last way we can look at claiming antibodies is by their method of production. For example, an antibody produced by immunization of a non-human with a peptide having a particular sequence. And this type of product by process claim is generally only allowable at the Patent Office if you cannot define the antibody by any other means. And in order to avoid a lack of clarity objection with this type of claim, you likely need to very specifically describe the immunogen. So it has 100% sequence identity. Again, so these last two methods are generally quite uncommon, and less than 5% of patents granted in the last few years define antibodies in this way via immunization or on the basis of a hybridoma. Looking at the requirement of sufficiency, the application as filed has to disclose at least one way of repeatedly producing antibodies having the claimed property. And the test for sufficiency at the EPO is whether it would be an undue burden for the skilled person to put the invention into effect. And claims defining the antibody by either its sequence or the antigen generally fulfill this requirement, but difficulties can arise when you define the antibody by way of its activity. So you would need to ensure that full details of the assay that you're using to measure activity are described in the application. And these requirements have to be satisfied at the filing date based on what is in the specification and what the skilled person would already know. And we can only use post-published documents for sufficiency if it's used to support positive findings in relation to the disclosure that's in the application at the time of filing. Another requirement of the Patent Office is unity of invention. And it can be that antibodies defined by sequences may be considered as separate inventions, which can lead to the necessity to file expensive divisional applications. That's unlikely to be the case if you're claiming all 6 CDRs, but if you're claiming, for example, only 3 CDRs and others are interchangeable, if you've got the data to demonstrate that, then all those different antibodies might be considered to be different inventions. And in order to avoid costs, you should try and include the particular novel and inventive features in the claim at the initial search stage. So if you're not claiming all 6 CDRs, for example, and including a functional feature, really want to make sure that's in claim 1 rather than thinking, oh, we might amend to claim 1. We might amend and include that feature further down the line. That might give you some unexpected costs. So it's really important to put the most important sequences first in the claims as well. And likewise, if you're thinking about using a method that's going to confer a particular inventive property, then you really want to consider having those features in the claim from the get-go so that you're not looking at increased costs. So in summary, for Europe, there are many ways to claim antibodies. And each one is likely to attract an inventive step objection, unfortunately. You can play devil's advocate a little bit with your specification, and you can, you really have to make sure that the full details of the unexpected advantages are included. If you know what the closest antibodies already are, it would be really ideal to include comparative data, indicating why your antibody is particularly better. Or evidence, for example, why isolating the antibody was to be unexpected. You really want to describe multiple properties and all the desirable features of the antibody from the get-go, since it can be difficult to rely on post-filed data at a later stage. It's not impossible, but ideally we want it all in the application as filed. To avoid any insufficiency objections, we want to make sure that the details of how the antibody was made, or the detail of its structure and function and relevant sequences, both of the antibody CDRs, for example, and any antigen or epitope are all included in your application on filing. And we can include multiple claim types and lots of different fallbacks to provide flexibility for amendment and argument during prosecution, but it all has to be in there when you file your application. So I think that's it from me, and I will hand over to Monique.
Thanks, Kirsty. Get my slides up here. All right, I hope you can all see my screen. So for the second section of the presentation, I'm going to be focusing a bit more on the actual scientific experiments and analysis that can be done to provide you with that relevant information for your patent. So to begin, I'm just going to go over a quick agenda. So just going to give you a bit of background on RapidNova, some of our services, as well as how you can use proteomic-based applications to help with your antibody claims and IP. So there are three main services that I'll be going through and discussing the technology, the requirements, and the applicability to filing your patent as well. So RapidNova is a proteomics-based company based in Canada. And our main mission is to elevate human health by decoding immunity. Now, how we're doing that is we're looking at proteins and antibodies that exist within the body that are present in databases, and we're looking at fine-tuning those. So we're shifting antibody R&D, and we're starting at the end with things that we know work, and then fine-tuning and engineering backwards. In terms of RapidNova's background, we've completed over 7,500 projects. We work with 18 of the top 20 pharmaceutical companies globally. And we were actually the first to sequence polyclonal antibodies using an only proteomic approach. So we're really pushing the frontier in terms of what you can determine and information you can obtain through a proteomics lens in a variety of different applications, and in this case, more specifically for patents. We've got over 20 plus years of innovation in the proteomics space from our scientists and founders. And currently, we've got over 13 mass specs in-house to help us do our proteomic work. And we're the largest privately owned proteomics facility in Canada. So I'm not going to spend too much time on this slide, but just as a brief background of our service offerings, the first two up there are revolving around our next generation protein sequencing service. So de novo protein sequencing, looking at a protein or antibody that exists and obtaining the amino acid sequence for both monoclonal and standard proteins, as well as polyclonal antibodies. And then we have generic proteomic services, which include more specifically, HDX-MS epitope mapping, as well as SPR Kinetic Analysis of binding molecules. In terms of claiming antibodies, so Kirsty mentioned 6 different approaches to how you can claim your antibody. And of the 6 that she mentioned, structure and function came up quite a bit, as well as target epitope. And those methods or approaches are all ways in which RapidNova can contribute to your patent. We've got very specific services that directly correlates to those attributes that you would be looking for to include in your patent. So for structure, we've got REMAb protein sequencing, where we can obtain a full-length antibody completely de novo. To contribute to function, we also have SPR Kinetic Analysis, where we can obtain screening data, on-off rates, binding affinity, and then to contribute to the target epitope approach, we can do HDX-MS epitope mapping for both linear and nonlinear conformations. So for all 3 of these technologies, I'll get into details around the technology, the requirements, and our specific advantage in how we do this work, as well as the IP applicability that goes along with it. So to begin with REMAb protein sequencing, I've provided you with a really high-level overview of the work that goes on. There's a lot more detail, but it would take me much longer to explain. So for the sake of time, we generally start off with a standard purified protein. we're going to be doing a multi-enzyme digest. Typically, we use between 5 to 10 different enzymes. And the reason being is the more variety in peptides that we can get, the better, the larger amount of data that we have, and the more holistic of an image we can obtain of what your sequence could be. So we'll put those peptides through our mass spec. We have a machine learning algorithm called Nover that was written that does the initial sequencing work. And then we have at least 2 bioinformatics specialists that will work with the algorithm and make sure that everything looks neat and tidy, and that we really convinced ourselves of the sequence and are 100% confident in it before we put it all together in a report for you and share that with you. In terms of requirements and timelines, so a standard project for sequencing takes 2 weeks. We can expedite this if you're in a rush. And as a standard, we ask for 100 micrograms. However, if you have a little bit less, we can talk, and there's a good chance we'll still be able to work with it. We can work with conjugated antibodies as well. And in terms of buffer, any standard buffer is fine. We like PBS. Additionally, if you've got a specific antibody from a unique species, or say you've done some engineering and it's a funky format, we can work with that as well. Having done quite a few of these sequencing projects, we are able to handle a variety of different curveballs that you could throw at us. And lastly, we also include isoleucine-leucine differentiation within this technology. So if you're not familiar, isoleucine and leucine have the same molecular weight, and it can be difficult for the mass spec to differentiate between the two. We were the first to develop a technology that we call WILD, which stands for double ion isoleucine-leucine differentiation. And with that, we're able to break isoleucine and leucine up into their W ions, and we can differentiate between the two. This is important for you because you don't want to have ambiguity in the CDR sequences that you're contributing or using for your patent, or even if you're doing the full protein sequence, that can lead to a lot of different variables that you'd rather avoid. In terms of IP applicability and rapid Nover specific advantage, having the protein sequence contributes to the structural features approach. So whether you're only including the 6 CDRs, or if you want to include the full monoclonal antibody sequence, because maybe the constant region of the antibody is important for some binding complex, we can do that. We are getting end-to-end sequence of the antibody, and we're getting 100% accuracy. So you want to make sure that the sequence that you're including in your patent is what you think it is, and it, if needed, becomes easier to defend down the line as well, when you got the right thing the first time and you don't need to go back and change anything. Additionally, there's no genomic ambiguity because we're coming at it from a proteomics approach. So, DNA sequencing is definitely a fantastic technology and has many applications. But when you're looking at sequencing an antibody that's present, there is that potential for extra heavy and light chains, which is quite prevalent, more so than expected. And so, you just want to be able to have that surety that the molecule that you're looking at is in fact what you think it is, and has a direct amino acid sequence with no extra bits and pieces or noise in the background there. And finally, if you have any novel formats of antibody, we can very likely work with it and still provide you with the sequence of it to include in your patent. So moving on to HDX-MS epitope mapping. So I put a little schematic diagram for you on the right-hand side there. Generally with HDX-MS, you're measuring the exposure of hydrogen molecules on the surface of your protein or your protein complex after quenching it with deuterium. So how this is done is you would incubate different fractions of your protein with D2O solution in a time series. And then through quenching, you control the temperature to prevent further exchange or back exchange. That complex is then digested, and then it's run through the mass spec. And those results are then analyzed and plotted. Now, this would be done for both the bound as well as the unbound complexes. And hydrogen molecules that are buried within the structure of your protein, for instance, at the binding site of your antibody antigen complex, are not going to be exchanged as readily. And so when you examine intact proteins and proteins via mass spec, and then you compare them with the amino acid sequence, it's possible to determine which region is engaged in binding. Some benefits to this method of epitope mapping is that you are able to obtain linear linear conformational and structural epitopes. So there's no ambiguity there. We do have specific mass spec to do this HDX-MS work. We use Bruker's. So we do have a very high degree of resolution here, and we're pretty fast at it. So speaking of timelines, we can do this work as a standard in 4 weeks, but we can bring that down, if you're in a time crunch. In terms of protein amounts, generally, we would ask for 150 micrograms of the antibody as well as the antigen. However, I put an approximate sign there because, depending on your protein size, we may actually need less, and maybe we'd ask for a little bit more. So it's worth having a conversation about just to be sure. In terms of buffer, 80% purity without detergents is what we would ask for. We are, using D2O and using a mass spec, so the purer the better. Again, we can work with any species or isotype, and if it is your antibody is conjugated, that's fine too. In terms of IP applicability and rapid NOVA's specific advantage, again, HDX-MS can contribute to your target epitope approach for your claim by providing you with information about linear and nonlinear epitopes itself. As Christy mentioned, you also want to prove that the results are reproducible. So, that's something that we can help you with as well. We've got the mass spec in-house. We have full reports detailing what we do, how we do it. So that kind of information can be included in the patent. And, if it needs to be redone, we also have our lab is still there. It's still in-house. So if you need to do any more analysis there, we can definitely help with that. As I mentioned, our results are highly specific, especially with the HDX-MS method of epitope mapping. We can get a really high level degree of quality with just a few amino acids on the sides for ambiguity. And with this method of epitope mapping and our specific HDX as well, we don't need the proteins to be labeled. So you are getting the natural functionality and binding of your proteins in question. So again, it's accurate, and there's no worry about potentially defending something that you didn't mean to in your patent application. Regarding our SPR Kinetic Analysis, again, I've included a little diagram for you there, but generally this is done by immobilizing a ligand. So in most cases, it would be your antibody to the metal surface of a sensor chip. And then you then inject your analyte or your antigen at a specific flow through rate to the system. And the concentration would be a specific one as well. After the equilibrium is reached within that system, you would inject a Reading buffer that would be continuously applied, and then the bound analyte would begin to disassociate. The disassociation is measured by the detection system, and then we can plot that on and provide some kinetic insight as to the on rate and the off rate, as well as the overall affinity of your analyte or the antigen to the ligand, your antibody. A few advantages of our SPR analysis.
Monique, you're frozen. Just wait for Monique to reestablish connection. Thank you for your patience.
Am I back now?
Yep, you're good.
Okay, great. So I'll just start from the SPR advantages things way. So regarding some of the advantages to our technology, this work is done in real time. So we can adjust our SPR settings to make sure that we're getting the best results, or the results that are the most noise free. Additionally, with the SPR that we're working with, it is label free. So again, you are getting accurate information as to how your antibody and antigen are binding, which would be useful, especially if you're looking for more data around affinity and looking to back your information up like that. We can also work with complex samples, and again, we work with high efficiency and accuracy as well. Regarding our requirements and timelines, we can take 2 weeks to do SPR. However, this can also be done in less time. And generally for SPR analysis, we ask for 100 micrograms of the antibody and the antigen. But again, I've got the approximate sign there because depending on the concentration of your sample, as well as the size of the potential proteins, those can all vary a little bit. We can also work with any standard buffer and any isotype. Regarding purity, we do ask for a higher purity sample because we are working with affinity, and we want to make sure that the reads that we're getting for your antigen and antibody are directly because of a relation between the two, rather than any extra noise or contaminants that could be present in the sample. So the cleaner, the better. Regarding SPR Kinetic Analysis, IP applicability and our specific advantage. So SPR contributes to the functionality component of the function approach to your patent, where we can provide you with information about on-off rates and the affinity of your molecule. Additionally, as Kirsty mentioned, if you're looking to provide information about newness, this is also a way to prove that your antibody is different than others that might be. Again, you need to also prove that your results are reproducible. So this is also done in-house. So we can also share how we've done this work, provide you with a full report detailing what you would need to include in your patent to prove that the affinity is what you expect it to be. Additionally, with our specific method, we are getting very specific data just because of the nanosensors that we're using. And another aspect that SPR is useful for is we can obtain the KD of your antibody and antigen complex. And then when doing epitope mapping, to set up an HDX-MS experiment, it's important to have the KD to be able to set the parameters correctly. So this can also contribute to HDX-MS as a natural next step if you were looking to include the epitopes as well as the affinity of a specific epitope in your patent application. So in conclusion, RapidNova provides quite a few services that directly contribute to ways in which you can file your patent for your antibody, detailing structure, target epitope function, as well as looking at providing novel aspects to your molecule of interest. With the REMAb, we're providing you with the full length monoclonal antibody sequence with 100% accuracy, regardless of whether it is a novel format, or if you're just looking for CDRs versus the full length. With HDX-MS epitope mapping, we can provide you with structural areas of importance, whether you want to detail those structural areas just by function or the full sequence of that specific area.
Frozen again, Monique.
Okay, am I back?
Yep.
Am I back now? Okay.
Yeah.
My Wi-Fi is not being friendly today. So with the SPR Kinetic Analysis, this is useful in ensuring that your molecule is can be backed up as being new with a better affinity, possibly. And all of this is done very highly specific and reproducible. So that concludes my part of the presentation, and I'll pass it over to Wei.
Great, thank you, everyone. So if you have any questions, please feel free to write them into the Q&A. I see there's a couple in here already, so I'll just start with that. First question is for Kirsty. In the context of a large antibody manufacturer who invents and manufactures thousands of unique antibodies, is there implied IP protection for these products, or do they need to be filed individually?
I'm afraid not. You would need to file patent applications. And I think that goes to the point that whilst you can generate lots of new and unique antibodies, as far as the patent office is concerned, you will have to demonstrate that those have advantageous properties above and beyond other antibodies that are already known. And when it comes to patent protection for technical inventions, there isn't any implied IP like you might have with trademarks or copyright, unfortunately.
Great, thank you. Next question is for Monique. Do you, Rapinova, also provide services for monoclonal antibody production?
So we don't produce antibodies, but we do have partners that we work with directly. So we can set that up for you if you're looking to do, a sequencing and expression project. But if it's just expression that you're looking to do, then I'd be happy to introduce you to some of our partners that we work with for the work.
Great. Next one is also for you, Monique. Is it possible to do HDX-MS epitope mapping for glycoproteins?
That is a trickier one. Glycoproteins do present a difficulty when it comes to HDX-MS. So there could be potential workarounds depending on the protein itself, but it definitely would be a harder project for us to take on. And we can chat about that with our HDX lead to get into the details if you'd like.
Great. There was one I answered in the chat. Just have some further clarification. So this one, Francois, if you're listening. So Francois, you mentioned that in HDX-MS, we say that the protein does not need to be labeled, but in the protocol, we describe needing labeling with D2O, and you are correct. Received some further clarification from our HDX lead in that what we mean by not requiring labeling is a covalent labeling dyes to monitor fluorescence. So D2O is a non-covalent label. Hopefully that helps to answer your question there. Next one. So is with regards to SPR, is the protein solubility required for protein sequencing and SPR characterization?
Is protein solubility required for SPR and sequencing?
Yes.
Is that the question? No, not for sequencing and for SPR. I think there are some other contingencies there. So that's a question about your specific protein that we could take to our SPR lead.
Great. Another one for SPR. What platform are you using for SPR? Is this an in-house derived instrument platform?
So we use Nicoya's software and instrumentation, and we have that in-house.
Next question is for Kirsty. For antibody drug conjugates, is a similar route to secure IP required for unique properties? for example, comparing to the native antibody?
Yes, so you would have to demonstrate that conjugating to the antibody was giving you something beneficial. So if the antibody is already known and you're conjugating it, you would have to demonstrate an improved effect. Likewise, if the conjugate, if the two parts particularly are already known and you are combining the two together, you would have to demonstrate a synergistic effect that was more beneficial than just using the two components individually.
Great. Two more questions here. This next one is for Monique. Could we do HDX-MS for antibody drug conjugates?
Yeah, we definitely can. We can work with complex molecules. So yeah, I'm happy to chat a bit more about your specific project.
All right, and the last one I see here on the chat, how affinity can be determined for oligomeric proteins consisting of identical subunits? Do you prepare Fab fragments for this experiment?
Yeah, I think that's a question more for our HDX lead scientist there and setting up how that project would continue on. So if I'm sure we'll get an email and we can have a future conversation about that.
Great. So I don't see any more questions in the chat, but everyone, thank you for joining today. If you do have any other follow-up questions or follow-ups to this webinar, please feel free to shoot an email to Rapnovor or to Monique, and we will be able to address those as soon as we receive them. So thank you, and another great webinar. Thank you, everyone.
Thank you.
Thanks. Bye.
Bye-bye.